Hello everyone, my name is Kevin Liu. I'm a Senior Marketing Specialist at GenScript. Today, I invited Dr. Liu Songying, who is the Group Leader of Antibody Discovery at our Antibody Production Department, to talk about anti-idiotype antibody generation and application in antibody drug discovery. Dr. Ying received his PhD from University of Massachusetts Medical School, specialized in antigen presentation and CD4 positive T-cell epitope selection. He then joined Pfizer as a postdoctoral fellow specialized in immunogenicity of therapeutic biologics. Dr. Ying has extensive experience in antibody drug development, especially in DNA immunization, high-throughput FACS screening, ELISA kit development, and anti-idiotype antibody development for antibody drug PD and PK studies. In this webinar, he will first give an overview of the different types of anti-IDs, then talk about their use in PK and PD, as well as immunogenicity assays. After that, he will review the obstacles and solutions surrounding the generation of highly sensitive, highly specific anti-IDs, followed by some examples from vaccine and antibody drug development case studies, which will wrap up Dr. Ying's presentation. Hope you can take this opportunity to learn from our expert and ask questions that you may have regarding anti-idiotype antibody. To ensure the sound quality, all the attendees are muted. Please feel free to type in your questions in the chat box in the lower right corner of your screen throughout the talk. Dr. Ying will address your questions together after the presentation. Without further ado, let's welcome Dr. Ying give his talk on anti-idiotype antibody generation and application in antibody drug discovery.
Thank you, Kevin, for the introduction, and thanks everyone for attending this webinar. As mentioned, I will talk about the generation and application of anti-idiotype antibodies in antibody drug discovery. This is the outline of my presentation. So I will first give an introduction to anti-idiotype antibody, and then focus on application and development of that. Following that, I will give several anti-idiotype antibody case studies, and I'm happy to answer your questions after that. So in order to set the stage for our discussion of anti-idiotype antibodies, I will first give a brief structural overview of antibody in general. On the left panel is a classic view of an IgG antibody. It consists of a light chain, as you can see with the green color, and a heavy chain, which is colored in blue. Both the light chain and the heavy chain have constant region, short as C region, and the variable region, short as V region. The V regions of light chain and heavy chain form the antigen binding site. On the right panel is a three-dimensional structure of an IgG antibody from the public domain with the C region, V region, and antigen binding site indicated. There are several types that we are always mentioning, and there is some confusion with those terms. Antibody isotype refers to the typical variations in the constant regions of the heavy and the light chains, as shown in solid dark on the right schematic view. Antibody isotype is species specific. For example, there are 5 isotypes in mammals, including IgG-A, IgD, IgE, IgG, and IgM. Instead, IgY is the major isotype in birds and reptiles. On the other hand, allotype is animal specific, which refers to the generically determined difference in antibodies between individuals in the same species, mainly a couple amino acid difference in the constant region, as shown as solid lines on the right. For example, mouse IgG1 from strain A differ from mouse IgG1 from the other strain B. They have the same isotype, which is IgG1, but different allotype because they from different strains. Last, antibody idiotype is antigen specific. referring to the antigen binding specificity defined by the distinctive sequence in the variable region of antibodies, as shown as solid dark in the right schematic view. For example, mouse IgG1 against antigen A shows those differences compared with mouse IgG1 from the same species, but against antigen B. They are the same isotype and allotype, which is IgG1 from the same mouse strain, but different idiotype because they recognize different antigens. I will next clarify several topes terms in anti-idiotype antibodies and lay the foundation of our discussion. The idiotope refers to the antigenic determinants in or close to the complementarity determined region, shown as CDR, in the variable region, as shown in the schematic view on the right. It can be the actual antigen binding site, and in some cases, it may comprise sequences within the variable region that lie outside of the antigen binding site on the antibody itself. Thus, it is possible for one antibody to have multiple idiotopes. So in the middle, paratope is a part of an antibody that recognizes an antigen in the antigen binding site. The counterpart of paratope is epitope, which is the part of the antigen binding to antibody. So what are anti-idiotype antibodies? They recognize the shared feature of idiotopes in the variable region of antibodies. At this point, I hope I have made the concept of anti-idiotype antibody clear. There are several different types of anti-idiotype antibodies, and mainly the three are shown here. The first one is antigen blocking or paratope specific, which inhibits the antigen binding when bound to the antibody drug and can only detect free antibody drug. The second one is non-blocking or non-paratope specific, which does not inhibit antigen binding and can detect total antibody drug, including free, partially bound, and fully bound. The third one is not that common, which is complex specific and can only detect bound drug. An important aspect of anti-idiotype antibodies is the ability to perform guided selection during the development process to isolate anti-idiotype antibodies with application-based specificities. Next, I will talk about the application of anti-idiotype antibodies. Anti-idiotype antibody is a must-have tool for antibody drug discovery. There are two major applications for anti-idiotype antibodies. It can be used to directly measure the antibody drug level in patient samples in pharmacokinetic assays, shown as PK assays, because they can recognize antibody drug specifically in high affinity. It can also be used as a positive control or reference standard for anti-drug antibody in immunogenicity assays, because anti-idiotype antibodies is also a kind of anti-drug antibody. I will expand these two applications in the next few slides. As you all know that antibody drug market is booming, with annual revenue close to $100 billion in 2015. In a recent review, among the 20 top-selling pharmaceutical products in 2013, half of them are antibody drugs, including the top three. Notably, only Humira and TNF antibody achieved $11 billion in 2013. One of the most important assays in drug discovery is PK assay. The main purpose of PK assay is to detect and quantitate human antibody drugs in serum. IgG is the most abundant antibody in human serum, making up 75%. Of that class, IgG1 makes up about 66%. The total serum concentration of the IgG1 subclass is between 5 to 9.5 mg per ml. The required sensitivity to detect antibody drug is in the nanogram per ml range, meaning assays must have ability to accurately detect antibody drug despite a million-fold excess of similar molecules. Agents with this degree of specificity include antigens or the drug target and anti-idiotype antibodies. Due to their different binding modes, anti-idiotype antibodies offer flexibility for specialized assay development. Here, I will reiterate the three main types of anti-idiotypes that can be useful in a variety of capacities in PK assays. Having both antigen-blocking and non-blocking anti-idiotype antibodies allows the quantification of free and bound antibody drugs in circulation, while complex specific anti-idiotypes illustrated on the far right can offer insight into the amount of bound antibody drug in serum. There are four typical PKS formats in ELISA-based. The first one is to use antigen to capture the antibody drug in patient samples and then detect with the secondary antibody. The disadvantage of this format is that coated antigens may alter the conformation to some extent, thus affecting binding affinities and the resultant assay sensitivity. All the other three formats involve using anti-idiotype antibodies to capture and detect antibody drug in the samples in sandwich or bridging assays. In Figure 2, the anti-idiotype bridging assay is an excellent example of using the same or two different anti-idiotype antibodies to detect and measure antibody drug based on the bivalent nature of antibody. For this type of assay, a highly specific anti-idiotype antibody is immobilized at low concentration and captures the drug in the serum sample. The bound drug is then quantified with a second labeled anti-idiotype antibody that binds to the free idiotype of the drug. This bridging assay is relatively difficult to set up as both arms of antibody drug may bind to immobilized anti-idiotype antibody, but this assay is very specific or is most specific. In Figure 3, the anti-idiotype capture sandwich ELISA is one of the most popular assay formats. In this assay, ELISA wells are first coated with anti-idiotype antibody as the capture antibody, then incubated with the antibody drug in serum. Detection is carried out using labeled anti-human secondary antibody. If one has both antigen-blocking and non-blocking anti-idiotype antibodies, one may quantitate the levels of antibody drug in the circulation, both free and bound forms. Figure 4 shows a slightly altered version of the bridging assay using labeled antigen to detect the antibody drug level, which is quite similar with Figure 2. Next, I will switch the gear to immunogenicity. Immunogenicity of therapeutic antibodies is caused by various product-related and patient-related risk factors, and it can cause different therapeutic outcomes in terms of drug efficacy and patient safety. Various methods have been established for measuring and predicting immunogenicity, and FDA has guidelines for risk-based mitigation efforts for immunogenicity. That is why we are all looking at this evaluate immunogenicity, and with this tool at idiotype antibodies, we can better evaluate immunogenicity. Here are some examples for immunogenicity incidence rate for several FDA-approved antibody drugs. In general, antibodies of murine origin, such as OKT3, have high immunogenicity. Chimeric and nice antibodies such as Rituxan and Herceptin show decrease, but still significant immunogenicity. Notably, fully human antibodies such as the bestseller Humira also indicate a certain degree of immunogenicity. This is why immunogenicity evaluation is required by drug administration agency for all therapeutic antibody discovery and post-market monitoring. Regarding the immunogenicity assay, due to the size of some clinical trials and the importance of testing patient samples at several time points, the FDA recommends a multidisciplinary approach. A rapid screening assay should be used initially, and any samples testing positive should then undergo a confirmatory assay, such as ligand or antigen competition assays. It is up to review experts to evaluate the sensitivity and the specificity of the assay itself and the characterization of immunogenicity. For example, whether anti-drug antibodies neutralize the activity of the product. Clinical reviewers must consider whether the immune response alters the serum levels of the product, affects clinical safety and efficacy, or both. Several aspects of immunogenicity assay development include high sensitivity and the ability to detect all isotopes. Other considerations include the avidity of the control used to evaluate the assay, as well as the effect of the matrix used to perform the assay. Rabbit or monkey will be the recommended host animals. In immunogenicity assay development, assay validation is a key step. Detection of anti-idiotype antibodies is the most important assay validation for immunogenicity. Negative control usually includes a pool of sera from 5 to 10 non-exposed individuals. This slide lays out the overall immunogenicity assay development, and the key is to use immunogenicity and idiotype antibodies to as positive control to validate the assay. For immunogenicity assay development, the most common assay format is the direct binding or bridging ELISA. In this type of assay, antibody drug is coated and the detection of anti-drug antibodies is done with labeled secondary antibody or antibody drug. For all immunogenicity assays, one must keep in mind that positive controls are necessary for proper assay development. If non-primate animals are immunized with a full-length antibody drug to develop a positive control, the antibody response will likely to be against the FC region or the constant region. Such a positive control may not be relevant for the anticipated immune response in human patients. Where the response to antibody drug is primarily located to the variable regions. Ideally, the anti-idiotype monoclonal or polyclonal is the best positive control for immunogenicity study. That is why we spend so much efforts, and also the field spends so much efforts in anti-idiotype antibody development. In addition to direct bridging ELISA assays, there are other assay formats for measuring immunogenicity, each having distinct advantages and disadvantages. Bridging ELISAs and radioimmunoprecipitation assays are sensitive, inexpensive, and typically require basic equipment. However, they may not be able to detect early immune responses and can be influenced by high levels of circulating drug. Conversely, surface plasma resonance, or as short as SPR, is the preferred method for detecting early immune response and has antibody characterization capabilities. Its utility, however, is hampered due to the expensive equipment required and high false positive rate compared with other formats. Finally, electrochemiluminescence assays, or ECL assays, have the advantage of sensitivity, but may lack ability to detect rapidly dissociating antibodies. Thus, assay choice depends largely on the stage of drug development and access to necessary equipment. Regardless of the method selected, positive control or reference antibody is needed for appropriate immunogenicity measurement. Anti-idiotype antibodies could serve this purpose. Purified anti-idiotype antibodies can be spiked into the matrix selected for screening assay and used as a reference standard. Next, I will talk about the development of anti-idiotype antibodies. We will dissect the obstacles and provide solutions which have allowed us at GenScript to have a 100% success rate in anti-idiotype antibody generation. So the first questions would be whether to develop polyclonal or monoclonal anti-idiotype antibodies. As mentioned, there are two major applications of anti-idiotype antibodies: directly measure antibody drug levels in PK studies and as a positive control for immunogenicity studies. With that in mind, polyclonal anti-idiotype antibodies are usually recommended for immunogenicity assay studies, as they are a heterogeneous population which mimic the real anti-drug antibodies incidence in human serum and are cost-effective. The drawback would be that they often embed batch-to-batch variation. On the other hand, monoclonal anti-idiotype antibodies are highly recommended for PK studies with the feature of homogeneous and defined specificity. For the same reason, it may not represent the real anti-drug antibody instance in human serum, and therefore it is not recommended for immunogenicity studies. But a combination of monoclonal anti-idiotype antibodies with distinct affinities can also be used for that purpose. Anti-idiotype antibodies are targeting the refined V region as the antigen binding site, which brings substantial challenges in anti-idiotype antibody development. To generate successful anti-idiotype monoclonal antibodies, appropriate immunization and haploidoma screening strategies are critical. For the generation of anti-idiotype polyclonal antibodies, the key factors are immunization and affinity purification. In terms of immunization, the CDR region of the antibody may not be very immunogenic, meaning that the antibody antigen may be unable to elicit an adequate immune response. Another obstacle to the generation of anti-idiotype antibodies is the fact that the percentage of anti-idiotype antibodies in hyperimmune anti-serum is very low. This can be compounded by a situation in which most of the antibodies raised target the Fc region of the antibody antigen. During the haploidoma screening process, an anti-idiotype clone, which coexists with a non-anti-idiotype clone, will be missed if conventional screening is applied. In the next few slides, I will dissect these obstacles and provide GenScript expertise for possible solutions. So to overcome the lack of antigenicity of the CDR region for anti-idiotype antibody generation, we can use an antibody drug KLH conjugate or FAB fragment as immunogen. The advantage of using FAB fragments is that constant regions which can induce unwanted antibody response in immunized animals are absent. At GenScript, we typically use full-length antibody for monoclonal anti-idiotype antibody production, and FAB or FAB2 fragments for polyclonal antibody production. The reason is that we have more screening in the monoclonal antibody production, and in the polyclonal there are only few steps for counter screening. Another method to circumvent the CDR region lacking immunogenicity is to adjust the immunization schedule or use our proprietary immunoadjuvant. In addition, GenScript has a broad range of animal hosts available to ensure optimal anti-idiotype antibody production conditions. So to overcome the issues surrounding the low percentage of anti-idiotype antibodies in serum, it is critical to use a proper secondary antibody, such as anti-mouse Assay secondary antibody, to prevent cross-reaction with human IgG, as most antibody drugs are humanized antibodies or fully human antibodies. In addition, high-throughput binding screening and capture ELISA assays can be used to select the natural epitope recognized by the antibody. In a capture ELISA, the target antibody antigen is lifted in solution instead of directly coating on the plate, in order to avoid epitope denature or masking. Selecting of the optimal anti-idiotype clone or clones from numerous anti-idiotype antibodies is perhaps the most critical step for successful anti-idiotype antibody development. Methods for detecting and monitoring anti-drug antibodies demand reagents and controls with high specificity and sensitivity, particularly when the drug is a monoclonal antibody. In fact, often a range of affinities is designed for certain types of assays, such as immunogenicity assays. At GenScript, a customer can choose the type of anti-idiotype antibody they would like based on their application. One of the first methods used to facilitate this process is epitope binding and antibody pairing. In this case, one anti-idiotype antibody is used to capture the drug, and the other anti-idiotype antibody is used to detect it. We will see a case study illustrating this in the next few slides. Next, affinity ranking may be useful in situations where a broad range of affinities is desired. For PK studies, high affinity anti-idiotype antibodies can increase assay sensitivity. However, for immunogenicity studies, validating the assay with anti-idiotypes of different affinities can prove the assay is able to detect anti-drug antibodies with different affinity ranges. In antigen blocking assays, anti-idiotype antibodies compete with antigen for drug binding. In PK studies, if customers have both antigen blocking and non-blocking anti-idiotype antibodies, they can quantitate the serum drug level both in antigen-free and antigen-bound states. This is a general flow chart for GenScript anti-idiotype antibody package. Both anti-idiotype monoclonal and polyclonal services are available. In the monoclonal package, we start with 2 to 3 mg of antibody drug and isotype control, and will deliver high specific hybridoma cell lines, supernatant and purified anti-idiotype antibodies. In the polyclonal package, the starting material would be 20 mg or more antibody drug and isotype control, and the deliverables include 0.5 to 3 mg specific purified anti-idiotype antibodies. So we can also include other customizations, such as whether you need anti-blocking or non-blocking antibodies, whether you need different levels of cross-absorption, for example, those isotype control or fully or total human IgG as the counter screening. So next, I will give a couple representative case studies of anti-idiotype antibody development. In the first case study, the target antibody is commercial anti-PD-1 antibody drug, Keytruda from Merck, and this is, I think this is one of the most famous antibody drugs nowadays. We are aiming to develop both antigen blocking and non-blocking anti-idiotype monoclonal antibody for PK studies. After all the positive and negative screening steps, we obtained more than 20 highly specific clones. We next evaluated whether these clones are antigen blocking or non-blocking. In the figure, we showed the antigen blocking results for 4 representative monoclonal antibodies. As you can see that binding of mAb1 and mAb2 to Keytruda cannot be blocked by the drug target PD-1, even at high concentrations. and therefore they are non-blocking anti-idiotype antibodies, which can be used to detect total antibody drug levels in PK studies. Instead, mAb3 and mAb4 showed dose-dependent inhibition by PD-1 and belong to antigen-blocking anti-idiotype antibodies, which can be used to detect antigen-free antibody drug levels in PK studies. We are currently developing ELISA kit for Keytruda PK studies. In the second case study, the target drug is humanized monoclonal antibody, and the application is to develop specific anti-idiotype monoclonal antibody for PK studies. As you can see from the figure that after the hybridoma screening steps, the final subclones we obtained showed high specificity that only recognize antibody drug but not isotype control and total IgG in human serum. And we have obtained more than 20 clones. We only show 10 representative here. So in the third case study, the target antibody is also humanized monoclonal antibody, and we aim to develop high affinity anti-idiotype monoclonal antibody for PK studies. After all those positive and negative screening steps, we finally obtained multiple specific subclones of high affinity with KDs in sub-nanomolar range, as you can see for clone 1 and clone 2 and clone 3. Clone 4 also has a high affinity, and this KD was measured by Biocor in SPR. So in the fourth case study, I showed an example of monoclonal anti-idiotype antibody pairing. This is to develop an antibody pair for PK studies of a humanized monoclonal antibody. After the positive and negative screening, we obtained 23 specific monoclones. Next, we want to find an antibody pair for following PK studies. We explored a checkboard format to evaluate the pairing capability of 5 anti-idiotype antibodies. Briefly, each of the antibody was used as capture antibody and paired with a built-in labeled detection antibody. As you can see here that mAb2 is able to pair with other 4 clones, which are highlighted in yellow. The best pair would be using mAb1 or mAb5 as the capture antibody, and built-in labeled mAb2 as the detection antibody, with the detection limit in nanogram per ml range. If you remember, this range is in the range of most antibody drugs in PK studies. So the conclusion is that mAb2 is able to pair with other 4 clones, mAb1, 3, 4, and 5. So all the previous 4 case studies I shared are monoclonal anti-idiotype antibodies for PK study. The last case study I want to share is for polyclonal anti-idiotype antibody for immunogenicity study. This antibody target is a fully human monoclonal antibody currently in phase 2 clinical trial. For this target, actually we also have developed monoclonal antibody and idiotype antibody for PK studies. Here, we showed that purified anti-idiotype polyclonal antibodies from both rabbits demonstrate specific binding to the antibody drug, not to isotype control. These polyclonal anti-idiotype antibodies are currently in downstream immunogenicity assay development. To wrap up today's webinar, I summarized here features of GenScript anti-idiotype antibody service. Our antibody experts here have extensive experience developing high affinity, high specificity anti-idiotype antibodies for use in PK and immunogenicity assay formats. Additional features of our anti-idiotype antibody platform include high-speed production with 2 to 3 months turnaround time, antigen blocking, epitope binding, and antibody pairing assay capabilities. And we also have both anti-idiotype monoclonal and polyclonal antibody packages. PK and immunogenicity assay development and a proven track record of 100% success rate for anti-idiotype antibody development is show our capabilities and expertise. We are excited to announce that our PK and immunogenicity kits for antibody drugs are coming soon. Now, at this time, I would like to thank you for attending this webinar. I hope you found the information useful, and I'm more than happy to answer your questions.
Thank you, Dr. Ying, for such an informative talk on anti-idiotype antibody generation and application. I surely learned a lot. I would also like to thank you all for attending this webinar. Now it comes to the Q&A session. Let's first take a look at the question number 1. So you said you have 100% success rate of developing anti-idiotype antibody. May I know how many projects have you taken?
Thank you for the question. Since 2009, we have successfully delivered more than 100 anti-idiotype monoclonal and polyclonal projects for PK and immunogenicity studies.
So question 2. Does anti-idiotype antibody work for evaluating antibody drug, not only in preclinical stage, but also in clinical stage?
That's a good question. The answer is yes. As shown in the case studies, we have several projects with the antibody drugs in phase 1 or phase 2 clinical trials, or even marketed drugs. We currently do not have the certification to directly work with clinical samples, but we can develop anti-idiotype antibodies and perform assay validation. Then we deliver the anti-idiotype antibodies and assay conditions to our clients who have the in-house capability for PK and the immunogenicity study of clinical samples.
So another question is, when doing PK assay of antibody drug, what are the differences of anti-idiotype capture sandwich and anti-idiotype bridging formats? Like their relative advantages and disadvantages?
So the major difference is the detection antibody. In the capture sandwich format, the detection antibody is labeled anti-human Assay secondary antibody. In the bridging assay, the detection antibody is labeled anti-idiotype antibody. The capture sandwich format is relatively easier to set up and validate compared with the bridging format. On the other hand, the bridging assay can reduce non-specific signal of anti-human Assay secondary antibody by using highly specific anti-idiotype antibody as the detection antibody.
Thank you for the answer. Next question is, based on your experience, which kind of anti-idiotype antibody is more common, antigen blocking or non-antigen blocking?
So based on our more than 100 anti-idiotype antibody projects, most of the monoclonal antibody in anti-idiotype antibody development are antigen blocking, which is probably due to that most of the idiotopes are located closely in the antigen binding sites, as those regions contain most sequence polymorphism.
Okay. So, is it possible to use monoclonal anti-idiotype antibodies for immunogenicity studies?
Actually, the answer is yes. Even though monoclonal antibody is homogeneous, combination of monoclonal anti-idiotype antibodies with different affinity range have been considered to use as the positive controls in immunogenicity assays by mimicking the real heterogeneous anti-drug antibodies instance in human serum. So you can use that for immunogenicity assays.
Okay, so that's basically all the questions we have got here. Thanks again for your attending.
Thank you.